+1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Question 1. You are studying the molecular basis of a neuromuscular disease of genetic origin. The major symptom of the disease is droopy eyelids, causing students to have difficulty studying for exams. You find that a specific muscle protein is poorly expressed in the eyelid cells of students suffering from the disease. You name this protein PSNOZ and the corresponding gene SNOZ1. For A, you wish to determine why PSNOZ is poorly expressed in the eyelid cells of affected individuals. Assuming you have a cDNA clone of the wild-type SNOZ1 gene and RNA samples isolated from the nuclear and cytoplasmic extracts of both wild-type and mutant cells, how would you determine if problem with expression is due to a defect in 1. Transcription, 2. RNA processing, or 3. Translation? Specifically describe the results you would expect and draw a schematic of the predicted data from your experiment for the following 3 possible explanations. 1. The gene is poorly transcribed. 2. There is a defect in mRNA processing. And 3. There is a defect in translation of the SNOZ1 mRNA. Nursingassignmenthelp.com. 1. To determine if the gene is poorly transcribed, you could perform a northern blot analysis and look at overall mRNA levels in the cells. You would run RNA purified from both mutant and wild-type cells. mRNA purified from nuclear fractions is probably best, on a denaturing polyacrylamide gel. Transfer the RNA to nitrocellulose and probe with a radiolabeled wild-type cDNA. If the mutant gene is poorly transcribed, you would see the result shown in gel 1. Another possibility would be to do an in vitro transcription reaction using the mutant and wild-type DNA, however you were not given the ingredients needed for this experiment. 1 point for naming the assay, 1 point for a complete explanation. 2. In order to determine whether the mutant is defective in mRNA processing, you could use a northern blot, see method in Part 1, to compare mRNA isolated from the cytoplasm and nucleus of both mutant and wild-type cells. mRNA isolated from the nucleus will be unprocessed and mRNA from the cytoplasm will be processed. Nursingassignmenthelp.com. By comparing the two side by side, you will be able to determine if the defective step occurs as the mRNA moves from the nucleus to the cytoplasm. Gel 2. 4 points, 1 point for naming the assay, 3 for explanation including proper controls. Another possibility would be to do an in vitro splicing reaction purified mRNA and ask whether the mutant mRNA is spliced properly. 3. If there is a defect in translation of the SNOZ mRNA, you would expect to see no obvious defects in the first 2 experiments. In order to definitively test whether there is a translation defect, you would want to purify antibodies to the protein and do a Western blot to determine the SNOZ protein levels in wild-type and mutant cells. Gel 3. 1 point for naming assay, 1 point for proper explanation. Nursingassignmenthelp.com. For B, you determine that the problem in PSNOZ expression is in some stage of RNA processing. Only a low level of the mature mRNA is generated in the mutant cells. To analyze the nature of this splicing defect, you decide to characterize splicesome assembly on the mutant SNOZ1 pre-mRNA. You have determined that the gene has only 2 exons. To your surprise, you find that all of the snRNPs, U1, U2, U4/U6 and U5, and U2AF interact with the mutant pre-mRNA at least as well as they do with the pre-mRNA from the wild-type gene. The assembled splicesomes, however, never generate active complexes or catalyze even the first step of splicing. Based on these results, in which of the splicing signals on the pre-mRNA, 5' splice site, branch site, polypyrimidine track, or 3' site, would you expect the mutation preventing splicing to lie? Justify your answer by suggesting a molecular explanation for the splicing defect. The SNOS mRNA is not processed properly in the mutant even though the snRNPs and U2AF can bind. There are two possible answers. The 5' splice site could be mutated, causing U1 to bind more tightly so that U6 is not able to compete effectively for the 5' splice site. If U6 cannot bind, then U2 cannot interact with U6 and the first step of the splicing reaction cannot occur. The second possibility is that the critical A residue in the branch site is mutated. This mutation does not preclude U2 binding but prevents the 2' OH from attacking the 5' splice site. NursingAssignmentHelp.com. There was a hint in the problem to help you avoid this model. A low level of mature mRNA is produced, but it was confusing since later it says that spliceosomes never generate active complexes. Credit was given for this answer even though it was not what we were looking for. To get full credit, you needed to have an answers as well as a clear explanation. For C, your lab has identified a drug that treats the SNOZ disease. You find that splicing of pre-mRNA is partially restored in cells from these treated individuals. What type of splicing factor might, if highly expressed, generate this recovered phenotype? Briefly justify your answer. You have now found that some mutant cells are able to recover and are asked to suggest a type of splicing factor that could rescue the splicing defect found in SNOS mutants. Using the first model from above, the dead box proteins that act as helicases and unwind RNA duplexes could suppress the tight binding of U1. By unwinding the RNA duplex formed by U1 and the 5' splice site, the dead box proteins would allow U6 to bind the 5' splice site and restore splicing. 2 points for a logical answer and 2 points for an explanation. 4D. Design a simple in vitro assay that could be used to purify such a factor from the recovered cells. NursingAssignmentHelp.com. You would want to use biochemical complementation to purify the factor that can suppress the SNOZ mutant. You would fractionate the extracts from restored cells and then add these fractions back to an in vitro assay. The most simple assay would be to set up a helicase assay in which you have an unlabeled mRNA and a small labeled fragment, representing the U1 RNA, hybridized to it. By adding back fractions and running the samples on a non-denaturing agarose gel, you could identify fractions that contain proteins that can unwind RNA/RNA duplexes and consequently cause the radiolabeled RNA to be released. Partial credit was given to logical experiments that did not show that the protein purified complemented the mutant phenotype. Question 2. You are working in a lab that studies differences in maternal and zygotic control of the fly embryo. In many multicellular organisms, mothers provide all of the proteins and RNAs required for the new organism to go through early embryonic development. Later in development, the zygote/new organism begins producing its own RNAs and proteins while the maternal ones are degraded. You are studying a gene called SOM1. Its predicted structure is shown below. The lines represent intronic or untranslated sequences, and the boxes represent exonic sequences. The nucleotide length is written below each segment. NursingAssignmentHelp.com. To look at expression of this gene, you decide to do a northern blot with cytoplasmic RNA from different tissues. You use a probe that will detect exon 1. The key indicates the origin of RNA in each lane. NursingAssignmentHelp.com.